Hi, good afternoon to all. So before we start, perhaps just let me have a quick check on the mic and also the session. So if you can hear me clearly and the session is smooth, just could you please help to type a yes to the chat box, please? Okay, I think it's all right. Thank you so much. So thank you for joining us for today's webinar. So we are going to have a session talking about the revolutionizing rare disease genomics with AI. And then before we start the sessions, could I have a few quick reminders? So we have a lot of participants joining the sessions here. So if you can help to mute the mics and also off the video throughout the sessions, that would be good. So let me now, okay. So now let me to have a minute to introduce our speaker for today, Dr. Vanisha. So Dr. Vanisha is already with us now. So she is the Vice President of Customer Success at Fabric Genomics. So she has the PhD from Queen Mary University of London, and then where she was trained under Professor David Van Hill, investigating the genetic basics of common autoimmune disease. So she leads the efforts to drive clinical adoptions, customer outcomes, and also growth across Fabric's genomics software platform. Drawing on her background in genomic research and clinical interpretation pipelines, she bridges the gaps between cutting-edge genomics technology and real-world implementation. Dr. Vanisha has been instrumental in Fabric's work with leading genomic center globally, driving software implementation, support, and training, and other activities across the most ambitious genomics projects in the field. So prior to her current role, she has been an analyst in Genomics England, where she evaluates clinical interpretation services and contributes to the variant analysis pipelines. So, um, so let's welcome Dr. Vanisha. Dr. Vanisha, the floor is all yours now.
Thank you. That's wonderful. Thank you for the wonderful introduction, and thank you for inviting me to speak today. I hope you all find the session useful, and I will now share my screen so we can make a start. Okay. Wonderful. So the title of today's webinar is Revolutionizing Rare Disease Genomics with AI. So that's what I will focus on today. And I don't need to give myself an intro, I got a good one, but I would like to introduce a little bit about the company I work for, Fabric Genomics, as I assume most of you may not be aware of what we do. So Fabric Genomics was founded originally as Omisha. That was the name back in early 2009. Between 2009 and sort of 2012, they really started pioneering the use of algorithms to understand variants better. And so one of the first awards that was given to Fabric Genomics was at the BioIT World Best Practices for an algorithm called VAST, which is published. And it was initially used as a research tool, and we had implemented it in our clinical platform along with a HPO-based tool as well. And then in 2015, they were selected as one of the partners for the Genomics England project in the United Kingdom, where Genomics England had set out to sequence 100,000 whole genomes as part of one of the world's largest clinical genome programs. I actually worked at Genomics England sort of in early 2016, which is where I got introduced to Omisha, and then I went over to work at Omisha to try and move forward their expansion into Europe. Also in 2016, they signed a pioneering contract, which really paved the way of the rest of how we developed our software with Rady Children's Hospital. And Rady Children's has a NICU program, and I'll speak a little bit about what they're doing in this webinar, but they were chosen as a primary genome interpretation partner for critically ill newborns as part of their NICU and PICU program. In 2017, we rebranded as Fabric Genomics and started expanding. And then in 2018, we got the Guinness World Record for the world's fastest genetic diagnosis with our partners at Rady. In 2021, we developed our first variant algorithm that built on the vast algorithm called Fabric Gem, which I will also speak about today. And that was published in a peer-reviewed paper. In late 2023, we did sign a partnership with Oxford Nanopore, and then last year we were actually acquired by GeneDx, which is a whole genome, whole exome reference laboratory in the US and is a market leader in that space. So we've spent 16 years building a platform that really makes genome interpretation faster, scalable, and actionable for those patients that cannot wait. And we really have focused on AI-powered interpretation for rare diseases. We do also do other applications, which I'll speak about in the webinar. So the acquisition by GeneDx that happened last year really brought the combined power of the GeneDx laboratory to Fabric Genomics and vice versa. Their mission is very much the same as ours, to diagnose patients quicker so they can live their healthiest lives through genomics. And with our AI tool and GeneDx's background of the last 25 years in the industry, we're really starting to find ways where we can support laboratories, clinical health systems, and hospitals to either support their in-house testing or even their send-out testing. And so we're happy to be part of the mission at GeneDx at Fabric Genomics. So now I'm going to get into more of the technicalities of what we actually provide at Fabric Genomics. We have a software as a service tool called Fabric Enterprise, which fits all the sort of industry standard architecture that you would expect for a software that's being validated for clinical use. And that's hosted on Amazon Web Services, allowing sort of that scalability and infrastructure. And we have AI technology within that software called Fabric Gem and ACE, which I'll speak about later in this webinar. The other part of our service, which is quite unique to what we offer, is called Fabric Clinical Services. And that is a team of clinical scientists and lab directors who actually take on the effort to do the analysis and interpretation for your cases in your lab, and then optionally can also provide a signed out clinical report by our lab director. So that particular team provides, as I said, interpretation services. They use the software, the Fabric Enterprise software, every day to sign out cases for our clients. And they are CLIA and CAP accredited, and they go through yearly proficiency testing. Our software and the work that Fabric Clinical Services do is all based on germline analysis, variant curation, interpretation, and reporting. And so that spans germline gene panels, exome sequencing, and genome sequencing. And that's true for short and long read sequences. So a little bit more about the work that we've done with ONT to enable a real scalable end-to-end workflow using the ONT sequencing with Fabric Enterprise software. So when we partnered with ONT, we really had this goal to bring rapid whole genome sequencing to the bedside, and we thought ONT was an excellent partner to enable that. And I'll speak about how others have also validated that that entire pipeline through peer-reviewed publications. And there's a couple of slides at the end of the webinar that I'll speak to about that. So as you probably know, ONT provide long-read sequencing, and they also provide base calling and variant calling through their Epitome platform. We have a solution where we can get the data directly from Epitome into Fabric Enterprise software for the downstream variant prioritization and classification using our AI tools. And then the final piece is the custom reporting. One of the advantages of having ONT as a partner is their project packs are structured as reagent purchases, where when you purchase the reagents, the instrument is also included. And this enables users to really get started quickly with a PromethION without any capital expenditure. And so that was a huge benefit to sort of how we partnered with them. And I'll speak to how some of the work that other projects have done with ONT as well in this webinar. And so one of the key advantages of using a technology like ONT with a software like Fabric Genomics, well, ONT provides real-time sequencing and real-time long-read resolution that you don't obviously you don't get with short-read sequencing. Advantages of long-read sequencing: there's superior detection of complex structural variants, repeat expansions, and of course phasing. Real-time generation is key for time-critical neonatal care. So you really want to get to those answers quickly. And so then having something like ONT validated in the lab enables you to do that. And there's, as I mentioned, a very low barrier to entry with adopting ONT sequencing with those PromethION project packs, which are included with the reagent purchase. So minimal capital commitment is required there. On our side, with the AI-driven interpretation, that automated prioritization by our algorithm GEM quickly ranks the likely causative variants across all variation types. And that's really key when you're trying to filter through thousands and thousands of variants that you typically get from exomes and genomes. You don't necessarily have that time when you need to get answers quickly. And so Fabric GEM is a really nice tool to speed up that process. And it provides the answers in a clinical context. So it's phenotype-driven ranking to really focus on the needle in the haystack. And then the VCF2 report functionality we have in our system means that there's no manual filtering required of any of the ONT data, or the ability or the need actually to set up any complex bioinformatics pipelines. We have all that in place within our tool. And then the final thing here in this slide is really with both of these, ONT and Fabric workflows, it really allows a decentralized clinical workflow. So what does that mean? That means that any lab can actually work to set up testing in-house quite quickly. So you can move from high complexity testing from central labs to the point of care. We provide end-to-end integration. So there's a seamless path from raw ONT data to a final customized clinical report. And it's scalable. So it's really designed to fit into existing laboratory frameworks. So now I'm really going to talk about the Fabric Enterprise software, what the pipeline looks like, and focusing on the AI tools so you have a good understanding of what our offering is. So our software, Fabric Enterprise, we really are describing this as a universal genome interpretation solution. Some of the key highlights I've listed here are it's an accurate software. So through the publication, which I'll describe in the next slide, the algorithm was able to call with accuracy 98% of diagnostic variants just in the top 10 prioritized list. And all that work is described in a key publication that was published in 2021. The application really provides speed with the GEM algorithm. Automated variant prioritization is covered with that. ACMG classification features are all built into the algorithm, and the final piece of clinical reporting. So there's an automated PDF generator in the system as well. Just to give you an idea of the amount of time it takes, a whole genome analysis takes about 1 hour, and it takes our clinical team maybe between 15 and 30 minutes to review that case and select the top candidates for classification. And then, of course, the lab director would have to do their final review. I've already mentioned scalability. We are a cloud-based platform, so we can support minimal amount of tests up to thousands of tests, and we offer customization at the report level. And then we do use industry standard security tools for encryption, and we comply with the regulations that are necessary. Focusing on sort of ONT, of course, you have the long-read sequencing pipeline, epitome analysis that covers base calling and variant calling, and then that all comes into the Fabric Enterprise software for the tertiary analysis part of the analysis. We do also have support for short-read sequencing, so just if there's any users in the audience that use short-read sequencers, we do support, of course, Illumina, Element, MGI, and Ultima. And with the long-read sequencing, we obviously do support ONT, which is the focus of the webinar today. Speaking a little bit about the tertiary analysis, so every single variant in the VCF from ONT will get annotated using our annotation pipeline. And then the GEM algorithm does the gene and variant prioritization, and we have the ACE tool that does the automated classification. And this will all make more sense when I describe those tools in the next few slides. We have a nice clinical reporting piece, and then systems integration is done via the REST API that 100% of our customers actually use to fully integrate their downstream and upstream systems with Fabric Enterprise. So that means data can come off the sequencer. You can set up an API to take that data directly into Fabric, and then set up APIs also to export data out of the system into your LIMS system, for example. Moving on to the GEM algorithm. So this is one of the core algorithms we have in our system that is AI-based, and it's really a gene prioritization algorithm, which considers multiple data sources to get to the answer quicker. GEM was published in 2021, and really what it does is think about, there may be people in the audience who have actually had to do a manual analysis of a whole genome patient or a whole exome patient, and putting in manual filtering protocols. Validating those protocols take a lot of time. You've got to validate sort of every filter, and then you still will have maybe 400 to 500 variants at the end of that list to still go through and actually determine whether any of those variants are causative for that patient. What GEM does really nicely is it actually takes the need of doing that manual filtering protocol, and it considers all these data models that I'm highlighting in this pictogram here. And this is typical of what a manual filtering protocol would cover anyway. So firstly, it looks at the variant effect and the frequency. So it's assessing, is this a loss of function variant? Is it a synonymous variant or a missense variant? And then what is the frequency of that variant in the general population? Is it rare? Is it common? And so it makes those assessments and derives a p-value from that genotype statistical model. The next piece of evidence it looks at is the patient phenotypes. And so what it does is it says, I have a set of HPO terms from this patient. How well do those HPO terms, how well are they associated with the terms in the HPO ontology at the gene level and at the disease level? So this audience may be aware that there is a full ontology of HPO terms that are used by multiple different vendors. The way that GEM uses that ontology is it's assessing the association of the HPO terms to the gene, but also to the disease associated with that gene. So it's doing it in a two-pronged method, which is really important because, of course, you want to know, do I have a good phenotypic match for that disease condition as well as the gene? It looks at sort of if there is a disease association, is that disease present in OMIM, and what's the inheritance pattern and everything else that goes with that OMIM condition? And then it also assesses, is that particular variant that's ranked in that gene, is that present in ClinVar? And what is the classification of that variant in ClinVar? Just to add here as well, with our, obviously, now our parent company is GeneDx. They've been in the industry for 25 years, and they have a very, very large gene curation database. So a lot of the genes they've curated, they've also curated the diseases for those genes. One of the pieces of work we've been doing this year is assessing, are there any novel gene disease associations that are not present in OMIM that we could input into GEM just to make the accuracy of GEM obviously improve the accuracy of GEM with these novel gene disease associations. And we did find a bunch of novel gene disease associations in the GeneDx database that are now available to us in our platform. And so this is one of the benefits of having GeneDx as our parent company, is we can utilize a lot of the curation information that they have as part of their database to enhance what we already have at Fabric as part of our algorithms. And then at the bottom here, you've got sort of the zygosity. So we will look at the zygosity of the of every single variant, and it will also look at whether there are large regions of homozygosity as well. It does a really good job of assessing the ancestry of the patient. It looks at the segregation as well of the data, and then it has an error model as well to assess variant quality. So we have GEM has its own error model for Illumina data. It also has its own error model for ONT data. So that really enhances kind of the quality of the information that GEM is ranking. And so going back to what I first said, with a manual filtering protocol, you do have to validate every step that you're taking. With GEM, you're just validating GEM, and that takes much less time. And through the benchmarking work that we did when we published the algorithm, what we found was when we benchmarked GEM on cases from Rady Children's Hospital, we found that by just looking in the top 10 ranked genes, GEM found 98% of the variants in that top 10 list. And so that is a huge differentiator when you're looking at getting to speed, getting to accuracy, and getting to the answers quicker. And then that sensitivity also obviously went down a little bit when you look at the top 5, top 2, top 1. But even when you're looking at the top just 2 genes, you're still getting mid-90s sensitivity. So that's really important. And that set of benchmarking data contained 120 diagnosed SNVs and 56 structural variants. So that's really key in understanding that GEM is not just looking at SNVs and those sort of easier variants. Is also looking at the most complex structural variants as well, and CNVs. And everything that I'm talking about here is in the paper, which was published in 2021 and can be found online. One of the, this slide I really wanted to show everybody here because with our partnership with Genomics England that we've had for many years, they also were very interested in GEM. And so last year, they wanted to test GEM on a set of 203 cases that had been processed through their clinical pipeline, but also their research pipeline. And so a bunch of these cases were easy to solve, and these cases had gone through their clinical pipeline, and a small percentage of the cases were their research cases. So the protocol at Genomics England is if there's a negative clinical case, it actually enters their research pipeline, where they have a team that perform manual analysis of those research cases to see if they can surface anything else for that particular patient. And what GEM did was it took all the data from those cases. They were processed with Illumina Dragon, but this is just to highlight the performance of GEM. And GEM prioritized all forms of variation. And you can see in this table here that the variant category spanned SNVs, short indels, CNVs, and complex structural variants. And the cases correctly prioritized by GEM, 97% of cases containing SNVs and short indels were correctly prioritized by GEM, and 100% of cases with those CNVs and SVs were correctly prioritized by GEM. And so one of the key take-home messages here is that GEM successfully prioritized 100% of the standard clinical cases, and actually 95% of the more difficult research cases. So even those difficult research cases, GEM still got to a 95% diagnostic rate. And those research cases contained a bunch of complex structural variants, cases where there was mosaicism, and cases where the phenotypes were very complex and pretty atypical. So it did a pretty good job there. So this is just to highlight that GEM doesn't just work on NICU data. We've also tested it on other validation cohorts in the paper, and more recently we've tested it with Genomics England data as well. And this data will be published this year. So let me show you what GEM really looks like and give you a snapshot of what all this looks like in our user interface. So GEM exposes the assessment of variants to the user, providing insight and transparency. So we're trying to give as much information to the user so they can also make an assessment of why this particular gene and variant was ranked highly by GEM. And so one of the nice things that GEM does is it outputs this score, which we call the GEM score. And basically, it's the log of the likelihood ratio. So the higher that GEM score, the more evidence there is in favor of that particular gene being involved in that patient's phenotype at that time. If there's a negative GEM score, it means that there is evidence against that particular gene being involved. And one of the nice things that we also surface up is all those individual component scores that contribute to this overall GEM score. And so we're looking here and we're displaying kind of what was the score for the OMIM disease match, what was the score for the phenotype match, looking at the HPO terms, what was the score for the variant quality. And so if all these scores are positive, that just means that all the scores have had a positive impact on the GEM score. If any one of these scores are negative, it means that that particular individual score has brought that GEM score down. And so it's really easy to understand if I have a lower GEM score, oh, it could be actually because the variant quality is not very good. And that could be because there's a skewed allelic balance or the coverage is low. And so it's quite easy to understand why GEM has ranked a particular variant in the way that it has. So it's surfacing up that really useful information. And then we have a lot of annotation as well, which I'll describe further in the next, in this slide. I wanted to show you a long read clinical case example that we ran through our platform. And so this is an affected female proband with generalized hypertonia and an intellectual disability phenotype. What we found was GEM had prioritized 2 candidates in this MCOLN1 gene that's associated with a condition called mucolipidosis. And you can see there's a canonical splice site on one allele as indicated by the phasing information that we get from long reads. And there's a deletion on the other allele. And you can see the phase information there. And that phasing information, of course, is unique to long read data. And it's really nice for the user to understand, which allele did this come from? And that just gives further insight into how they would interpret that particular variant without the need of actually having to run parents as well. And so what we're showing here is how did GEM score that particular gene? What were the ranked variants for that gene? And what's the predicted classification of that particular those set of variants according to the ACMG guidelines. One feature I wanted to show you here is also, I'm going to just do that again. When in the user interface, when a user hovers over the gene, the phenotypes change color depending on how well those phenotypes are associated with the gene. So again, that's another level of understanding for the user of the phenotypic association at the gene level. And so here you can see that MCOLN1 has a good association with hypertonia, corneal opacity. The association with intellectual disability is a little less, but it's still associated to that gene within that ontology tree that we use in the HPO. And the colored bars here indicate the ACMG classifications. It's very easy for the user to understand how did this variant classify according to either the ACE algorithm, or maybe this variant was already part of the database because it had been seen before in another case and it was classified as pathogenic. So we're surfacing all the previously curated information up as well for that particular variant. I've already mentioned the GEM score and the phase information, and here you can also have access to the gene disease condition. Lots of other annotations here also at sort of the variant level. So we're focusing on NORMAD 4.1. We have link outs to ClinVar, lots of in silico prediction tools, and then what we do surface up in the next phase of this analysis is the ability to review the ACMG criteria, and then the ability to also review some other annotations at the variant level, and then the ability to add that variant to the report for final reporting. And so at this stage, I can easily see that this particular variant was already classified on October 8th last year as pathogenic, and these are all the criteria that were invoked for that classification. I can see the variant inheritance as well, and I can see some variant quality metrics as well as the automated variant description. We also have link outs to Google Scholar, Splice AI, ClinVar, as I mentioned, and there's also a transcript table, some more Splice prediction tools, and again, the ability to really review the ACMG criteria for final classification and reporting. One thing to mention here as well is that think of GEM as a decision support tool. So GEM is giving the user what it thinks are a list of candidate variants. And it's up to the user then to evaluate those variants and decide whether they want to take any of those variants forward for classification. At the classification stage, we then provide predicted ACMG classifications according to our ACE algorithm. And those classifications still then need to be reviewed by a person for the final sort of classification application. So it's not a black box. You know, what we're trying to do here is help the user get to the answers quicker, help the user get to the classification quicker so they can move on and focus on other cases as well. A little bit about our ACE algorithm. So ACE was actually first developed for our hereditary panel pipeline. And what it does is it basically gives the user a predicted ACMG classification according to what the curation of that information is. So just let me explain a little bit more. ACE has a set of curated genes. So far, we've curated about 4,500 genes where ACE can give the user a predicted classification for any of the variants within those genes. ACE is basically a set of codified guidelines. So what we did is we codified most of the ACMG criteria. So it gets to either a benign, likely benign, pathogenic, or likely pathogenic classification. And it also predicts if a variant is VUS. One of the benefits of ACE is we do apply gene level cutoffs because we have all of the ACE genes in our database are curated by our clinical team. So our users have full access to that curated data and they'll be able to see what gene level cutoffs we've applied. So it's actually a very accurate classification algorithm. It doesn't overcall VUSs as pathogenics or pathogenics as benigns. It's actually pretty accurate. And as I said, it allows the user to understand which variants should I focus on first, especially in a panel workflow where you do have to look at every variant annotated to the panel. It allows the user to really focus on those pathogenic variants first, and then they go down the list and say, okay, now let me look at the VUSs, and then they may decide, actually, I don't need to look at the benigns or the likely benigns because ACE has done a pretty good job of classifying those variants. So it enables the users to really facilitate a faster and more efficient workflow. And one of the nice things that we have in the system as well is called a classified variant database. So as our users are classifying variants in their workspace, it's building their own curated database, and that knowledge database is really useful to understand, how many variants have been classified for a particular phenotype or for a particular panel or for a particular set of patients, such as NICU patients. And so that curated database is continuously being added to as more cases are being classified in the system, and it really enables the user to benefit, to leverage the large amounts of data that they're curating, enabling just that higher quality, more accurate diagnosis and automation. It also enables a VUS resolution. So if you had a particular variant that had been classified as a VUS maybe 6 months ago, maybe a year ago, and then we've made some updates to the application, maybe there's new database evidence or some algorithmic updates, the user will easily be able to see that if that variant had been classified, had a change in classification in a future case, just at the case level. And so that having access to that information is quite important. And then the final piece is the customization of the report. So this is a draft version of the PDF report that's automatically generated from the system, and it enables our users to really get to reports quickly. All the information here can be automatically added, and the idea is that then a lab director would review this report, would review the results of the report, and then perform sign out directly within the system. So we do support electronic signature in our application as well. A lot of features around the reporting as well. We support bulk approvals. We support the ability to retract reports, correct them, and then have new versions of the same report. And then, of course, we do offer customization. So we can add your logos, we can change the colors of the report, we can add customized fields, etc. So it's pretty flexible. I've now gone through sort of, what we offer at the software level. I've talked about Fabric Gem and Fabric Ace and the reporting piece. I now want to focus on a couple of customer stories who have used actually ONT sequencing and Fabric for some of the initiatives that they have done in their own countries. And just to introduce these stories, I wanted just to talk about some of the work that's been done by Rady Children's Hospital to really understand the need for rapid whole genome diagnosis, particularly in the NICU. And so Rady Children's Hospital have been pioneers in really understanding, if a baby is diagnosed quickly in the NICU, what are the effects of that on the hospital, the patient, and the entire health system? And so here, what I'm trying to describe is 44 studies of children in ICUs with diseases of unknown etiology, so typically rare diseases. 37% of those children received a genetic diagnosis, and 26% had consequent changes in management of their health. And because of those percentages, the healthcare costs were reduced by almost $15,000 per child because they had initiated rapid genome testing at the NICU level. There's also, we have rapid whole genome testing. We also have ultra rapid genome testing, which has a very low turnaround time. And so, of course, ultra rapid genome testing outperformed rapid whole genome and even rapid whole exome testing with a faster time to diagnosis. And it actually gave a higher rate to diagnosis. So if you test earlier, when those symptoms appear, the chance of getting a diagnosis is typically higher. And there is sufficient evidence through this work to conclude that ultra-rapid, or even rapid exome sequencing should be considered in all children with diseases of uncertain etiology at ICU admission. But right now, those tests are currently being performed only in less than 5% of approximately 200,000 children that are likely to benefit from this test. And these stats are all from the USA. Again, whole genome sequencing, we have proven through Rady Children's that it does reduce cost and improve care in the NICU. And a couple of studies done by Rady Children's and also with Project Baby Bear, excuse me. Oh, okay. Is that when you reduce the turnaround time, the cost savings for the system is much, much higher. So you've got a 3-day turnaround time. The cost savings for the system is around 2.9 million, and the cost savings for the child is a maximum around $15,000. And that cost savings obviously increases or decreases with the higher turnaround time there. So still, pretty good at a 14-day turnaround time. But again, you've got almost a 50% increase in cost savings if you reduce that turnaround time from 14 days to 3 days. So that's really, really important. And then on this side, I'm showing a table where it shows, again, if you reduce that turnaround time for getting a whole genome test performed to 3.6 days, in this study size that was performed, I think this was Project Baby Bear, they saw a 49% diagnostic rate and a changing in healthcare management at 58%. And that's obviously, much more than a 15-day turnaround time for an exome or a genome. Okay. Oh, excuse me. Bit of technical issue here. Let me go to the... Sorry, I'm just going to stop presenting and re-present again. Excuse me, sorry about that. Okay. Okay, here we go. Okay. One of the key initiatives now, just now you've got that understanding of sort of the cost savings and why whole genome sequences is really important, is now flipping now to how some of these initiatives have used ONT sequencing to really get to that cost savings as well. So BadgerSeq is an initiative that has been performed by Stephen Main, who is the director of human genomics and precision medicine at the University of Wisconsin in Madison. And BadgerSeq was really there as a goal to provide rapid genetic testing to infants in NICUs across the state, particularly outside major academic centers. And the impact of BadgerSeq was to reduce hospital stays, lower costs, and improve care by enabling faster diagnoses. And so the goal for BadgerSeq at a high level was to reduce that turnaround time to 3 or 4 days. And one of the goals also was to have an on-demand in-house sequencing for a moderately sized NICU. So one of the things that they said was, you know, a NovoSeq just didn't really fit what they wanted. They needed on-demand sequencing. If they'd done BadgerSeq with a NovoSeq, they would have had to fill up these flow cells, which then the sequencing turnaround time would have been a lot longer. However, with the PromethION solution, it's a very suitable alternative to a NovoSeq because they can just they can use 1 flow cell per patient or 3 flow cells if it's a trio. And then they decided to use Fabric Gem or Fabric Enterprise and the Gem algorithm as their tertiary analysis solution, and which enabled them to get to those answers really quickly. And then at the workflow level, just to highlight some stats here on the time it took, at the wet lab level, the DNA extraction and the library prep, where they used 2 flow cells per patient for the whole genome. And then that included base calling, methylation, and alignment on the fly. That took about 36 hours. Clinical interpretation with Fabric Gem. So the analysis and the actual interpretation of the data took about 2 hours. But then the final review and the report still took 2 days. So it still took somebody a little bit more time to review that report, perform sort of kind of a clinical review, understanding the data, and getting that report out took 2 days. So in total, this did take about 3 days for their turnaround time. And that all the information presented here is also available on the Nanopore website. So just to describe the initial results here, so they ran 9 trios with 3 positive controls. Nicely, all 3 positives were detected and 6 still remained inconclusive. So these are the positive controls. And you'll notice here that the controls consisted of SNVs, but also this deletion on chromosome 15, which is associated with Prader-Willi syndrome. And then we have a positive control. This is a compound Het variant at PMMM2, which is associated with glycosylation, and then a positive SNV in the gene EMC1 associated with a cerebellar atrophy. So initial conclusions from the BadgerSeq study, which used ONT sequencing and Fabric Enterprise, was the overall goal of 48 hours from DNA to initial variant analysis is really realistic for hospitals or clinical systems. The ONT PromethION is well suited for a moderate volume, ultra rapid whole genome NICU unit. Fabric Enterprise software is a really good fit for BadgerSeq because the rapid AI assisted analysis is essential for achieving BadgerSeq's timeline goals. And then the ability to analyze SVs and CNVs provide evidence for ranking are really clear strengths in using a tool like Fabric Enterprise here. The second and final customer story I want to describe today is actually from the University of Auckland, so it's a bit closer to you guys in New Zealand, and they wanted to set out to benchmark ONT and rapid genomics, and they did a feasibility study. So they validated ONT and is it suited for rapid genomics at their hospital in New Zealand. So the objective here was to establish and validate a scalable rapid genomic testing pipeline for critically ill infants using ONT and Fabric Gem AI-driven analysis. I'm showing the workflow here. So obviously this started from DNA prep all the way to base calling, where they generated the BAM files, and those BAM files went through different ONT calling pipelines to generate the final VCS that then went through to Fabric Enterprise, and they also did some benchmarking as well. So one of the key things they did in this study was they had a dual validation framework. So the first benchmarking framework was analytical. They used the Genome in a Bottle HG002 truth dataset to understand the precision and recall using the GA4-GH benchmarking framework. And then secondly, they also wanted to understand the clinical concordance. So they they performed a head-to-head comparison of this workflow with an accredited Illumina short read workflow. And through this benchmarking, they did achieve 100% diagnostic agreement across all 10 acute pediatric trio cases, which I'm describing in the next couple of slides. So just to go through the timeline of this workflow before I present the final conclusions, this was a rapid trio genome workflow. So they had 10 trios. The ONT sequencing took about 24 hours. GEM analysis took about 1 hour, and then the clinical review and diagnosis took about 60 hours. So the median end-to-end turnaround time was about 85.5 hours. And the results and conclusions, so this was an integrated workflow, as I said, ONT sequencing, GEM analysis, and clinical review. The head-to-head validation comparing this workflow with Illumina short read was they found 100% concordance in the genomic findings. So out of the 10 trio cases, they had 6 positive diagnoses and 4 negative cases. So that pipeline demonstrated perfect agreement with an already established accredited clinical standard in all 10 cases. And some of the key findings here that Fabric GEM served as a primary platform for variant interpretation and prioritization, effective for SNVs, indels, and complex SVs. And one of the key findings is that we were able to uncover through the benefits of long read sequencing a complex GBA phenotype. So this is a 55 base pair deletion and a missense variant. And so that successful phasing information from ONT sequencing enabled us to see that information in the platform, really providing that advantage of long read data. And here is just a comment from Justin O'Sullivan. He was the main author of this paper, saying the concordance of genomic findings in both positive and negative cases demonstrates the applicability and reliability of our pipeline for acute clinical care. So these stories are really there to help you understand that ONT platform, along with our tool Fabric Enterprise, does allow great implementation of AI into genomics workflows with that speed and accuracy that's needed for some of these initiatives at the hospital level. And it also allows users to think about how would they apply this type of workflow in their hospitals by understanding how other initiatives have done that. ONT allows on-demand sequencing, enabling the implementation of rapid next-generation sequencing at the bedside with low capital expenditure. And then Fabric and ONT we have created an integrated solution that is affordable, fast, and flexible. And I'll just leave you with this final slide of why Fabric Genomics could be considered as your partner of choice as you're listening to this. We have proven our technology at scale, so we've been trusted by leading programs such as Genomics England, Rady Children's Hospital, and we've worked with a lot of other hospitals and commercial labs throughout the years. We do have a best-in-class AI with that 98% accuracy in the top-ranked genes. Gen gives the user confidence. It's an explainable AI, and it may also surface up variants that may have been missed by manual filtering protocols. So it's really nice that we have something that allows clinicians and users of our platform to feel confident in the results. We do have an end-to-end platform, so it's seamless workflow from raw data to interpretation to clinical reports. We have a fast turnaround time, so our whole genomes with the GEM analysis take about 1 hour, and it's clinical grade ready. We have been validated in many CLIA and CAP aligned workflows because we have sort of all those tools in the system to allow laboratories to validate quite quickly. Flexible deployment, so we're cloud-based. We're on AWS right now. We can deploy in any AWS in your region, and so please reach out if you feel like you want to discuss that with Stephanie, who's also on this call, and myself. And then remember, we also have the software and the expert clinical interpretation hybrid. So we have a fully clinical interpretation team in our business that can take that resource from you if you don't have it in-house to perform the interpretation of your data and provide you fully signed out reports under our accredited CLIA and CAP laboratory. We've got a continuously learning variant database that also improves accuracy over time. And so I hope that I've given you a good indication and outlook of what Fabric Genomics does and some of the use cases. And now I'm happy to answer any of your questions.
Thank you so much, Dr. Vanisha. It's really impressive. So actually, I have done some rare disease projects before, so it really take very long time, starting from the sequencing data all the way identify the variants, and then prioritize the variants and identify the genes related to the case. It takes very, very long time, but now it seems like it's so easy. Okay.
Yeah.
I think I saw a few questions from the audience, so perhaps I can pass the questions to you. I think the first question was, what is the range of the GEM score? I think it's related to this score that you mentioned just now. I think 1.7, 1.8 in your examples that you mentioned.
That's a wonderful question. Yes. So GEM, the score, just to recap, GEM outputs base factors. And so that score essentially is the likelihood, is the log of the likelihood ratio. And so the highest score I've seen is about 4.5. To translate this, if you have a likelihood ratio of if you have a likelihood ratio of 10, then the GEM score will be 1. If you have a likelihood ratio of 100, the GEM score will be 2. So a likelihood ratio of 100 means there's 100 times more evidence that gene is involved in that patient. A likelihood ratio of 1,000 means there's 1,000 times more evidence. So that's the way to translate it. So if you have a GEM score of 4 or 4.5, that's maybe like a slam dunk variant. That is definitely a candidate that you would want to consider to move forward. And then the negative GEM scores, of course, they go down to sort of, I've seen GEM scores of like -1, -2. Probably that's the max. And the negative GEM score just means that one of those individual component scores is decreasing that score because it's had a negative impact.
Thank you. And the next question, I think, is more on the recording, whether we can share the recording. So most of the time we will be able to share the recordings. Of course, we will be getting the consensus from Fabric Genomics as well, because the recordings will have all the content shared by Fabric Genomics and GDX. And the next question will be, the data comes from different dimensions like genetics, clinical information, zygosity. So in all the algorithms, are all these dimensions analyzed separately or together? If together, how are the weights determined?
Yeah, so a GEM is a probabilistic algorithm. So it's looking at, if you remember that pictogram, all the different data models actually are priors to the underlying GEM model. So those priors are then determined in the probabilistic model and then weighted. And then the individual weights of those priors then are then defined by the final likelihood ratio. So the beauty of GEM is because it has this probabilistic nature in the model and it has these weighted priors, any one of those individual inputs can have an effect on the score. And that's kind of opposite to what a deterministic model would look like. So you may have a situation where actually the variant quality is low, but because the weighted prior for the phenotypic match is really high, you will have like a positive GEM score, because GEM wants you to know, although the variant quality is a little bit low here, there's a really good phenotypic match. and you may want to consider this. And if you the user or the clinician, whoever is analyzing the data, believes that that particular variant and that gene actually probably could be causative, it's then up to you to go and then determine if that variant is real by a confirmatory method. And so that's kind of the nice thing about GEM is that probabilistic nature. It has those weighted priors to determine if that particular gene is really going to be considered, and it takes all those different evidences into account in those priors. Hopefully that makes sense. But the paper actually will detail this in a much more eloquent language than I have done, but it will give you much more information on how GEM is actually doing the assessment.
I see. So I think we have more question. What is a VUS resolution indicate exactly?
Yeah. So one of the key things in clinical laboratories, and this is a lot of work has been done by ClinGen, which Heidi Wien is a part of, is how to report VUSs in the clinic. And so VUSs, if a patient that gets a VUS report, it's quite hard to understand what to do with that. So what we're trying to do with our software is, because every time a variant is classified in a workspace, and a workspace belongs to a user, it's stored in that database. So if you had a VUS in patient A 6 months ago, today with new pipeline evidences, new databases, that VUS actually may now be a likely pathogenic variant. And so if that likely pathogenic variant is seen in another case, you'll be able to see, oh, actually, it was classified 6 months ago as a VUS in patient A, but now in patient B, the classification is upgraded to likely pathogenic. And so having that level of information and understanding, do I need to now go and reclassify my VUS based on what I know about this patient and upgrade it to likely pathogenic and reissue a new report? And so, VUS resolution is actually quite important from a clinical perspective to really give the patient a better understanding of whether their initial VUS actually could now be upgraded. And that the upgrade of that classification could be because there's actually new literature information, because there's new functional evidence supporting that. Maybe there's more families that now have this particular disease with this variant. So all that evidence is sort of, taken into account when you're doing VUS reclassification.
I see. So I think next question, how does Fabric Gem handle phenotypic data? So if they have the very sparse HPO, I think it's the human phenotype ontology terms. So how that impact the AI prioritizations?
Yeah, so GEM uses the HPO terms as Bayesian phenotypic priors. So I kind of alluded to this in my other answer. And what it does is it reasons over the ontology relationships rather than exact term matching. And so it has a lot of knowledge on those ontology relationships from the HPO. And actually the paper, it describes this pretty well. So if the clinician provides really sparse manual HPO terms, it's still supported with strong gene prioritization. But actually, the richer phenotypes, if you get richer phenotypes from the clinician, it just improves the GEM score a little bit. But even with sparser phenotypic terms, GEM is doing a pretty good job. And so that's all described in the paper as well.
I see. So I think there's another question. I think this one from one of my colleagues. So is there a documented use case for using the Fabric platform to analyze genomic or clinical data for FSHD detection?
I'll have to ask my clinical team. They may have been aware of some of these cases in their pipeline, and so I can definitely go back and ask if they've looked at any cases with that particular phenotype.
Sure. And then we have questions on the Badger-seq workflows, I think in one of the case studies. So it targets a 48-hour DNA to analysis goal. In practice, what is usually the biggest bottleneck to hitting the under 2-day time frame? The wet lab, the sequencing, or the clinical review?
Yeah, that's a good question. So technically, with the ONT and Fabric pipeline, we have resolved the data bottleneck. For now, actually, the bottleneck in practice is probably the human and logistical overhead. So it's probably going to be the time taken that sample gets to the lab, the wet lab preparation of that sample, and then on the other side, the actual review of the final data. So the clinical MDT that's performed between clinicians to really decide amongst themselves that, yes, this is the answer for the patient. So the data bottleneck, I feel, is we're getting to a place where that's being resolved. It's sort of a human logistical bottleneck that's probably the largest time resource right now.
I see. And another question also from our side. I think I've asked this question also yesterday. Is there a trial token or free trial available to test the Fabric software? So,
Yes, of course, we're happy to have conversations with any of your users or clients on this webinar if they're interested in moving forward. We can give a more deep dive demonstration of the actual platform, and we can also provide access to a sandbox workspace so users can see what the data looks like. And then, of course, if users want to upload their own data and have a trial, we're happy to explore that further as well.
yeah, I think Malaysia here we do have some groups working on the rare disease, and then some of them already have some data. I think they are quite eager to try out.
Wonderful.
Yeah, so I think at the moment I don't see any new questions. Thank you so much, everyone. Have a nice day.
Thank you. Bye.